Hey everyone, Paul DiLuigi here, and tonight I have another video presentation for you, which will conclude our portfolio series. Tonight's portfolio series is going to feature the reproductive system, and the topic of discussion will be the regulation of sperm maturation in the germinal epithelium of the testes. So if we move along to our slides here, we start with our title page and into our first slide. So before we get into the genesis of sperm cell production, let's take a quick look at some of the anatomical structures that are related to this process. And we're going to start with the testes. The testes are located in the scrotum in the inguinal region of the male body. They're oval in shape, and they're also made up of 1,000 seminiferous tubules. In between these seminiferous tubules, there is a dense layer of irregular connective tissue, which houses other important supportive cells, most importantly, the Leydig cells. Now, Leydig cells are testosterone-producing cells, which is an essential hormone in spermatogenesis, and they also have conversion capabilities, which change testosterone into estradiol. Moving along. So as stated in slide 1, the seminiferous tubules is where the genesis of sperm reproduction begins. Within these seminiferous tubules, there are two types of cells which we can find. The first one is the spermatogonia cells, which give way to spermatids, and the supportive Sertoli cells, which help or nurture these spermatogonia cells. The Sertoli cells line the basal lamina of the seminiferous tubules, and they protrude apically towards the lumen of the tubules. The Sertoli cells also form a blood-testes barrier on the lateral borders as they nurture the spermagonia cells. The blood-testes barrier are made of tight junctions, and they limit or restrict the diffusion of molecules between the Sertoli cells, and they also limit the interactions of the spermatogonia between one another. If we move again, but backtracking a little bit to our second slide, Sertoli cells also provide nourishment, and they're responsible for the secretion of inhibin, actin, growth factors, enzymes, and ABP, or androgen binding proteins. All of those molecules are important in the regulation and the growth of the spermatogonia cells. All right, slide 3. The maturation process begins with some sperm cells undergoing the replication process of mitosis, adjacent to the basal lamina. Once mitosis is complete, the cell is now a germ cell or a primary spermatocyte, which is also a diploid cell. Once a primary spermatocyte has replicated, it is then replicated again, or it separates into a secondary spermatocyte, and this cell is now a haploid cell, meaning it only has half the chromosomes responsible, or half of the genetic material which make up a full complete cell. As this happens, the spermatid, or these secondary spermatocytes, replicate apically towards the lumen, and eventually they give way to a spermatid, or they transform into a spermatid through replication. Spermatids will remain embedded in the apical membrane of the Sertoli cells until they lose their cytoplasm and develop flagella, which is a means of locomotion, and this is the way they make way to the oocyte within the female body. Also during this time, the spermatids will also condense their chromatin within the head of the cell, and they will develop an acrosome, which is a modified version of a Golgi apparatus. And this modified Golgi apparatus houses important enzymes, which will be important in the fertilization process as it makes way into the female body and to the oocyte. When mature enough, the spermatid cells will release themselves from the Sertoli cells, and then they will be transported via bulk flow to the epididymis, which is another structure located within the testes, and this is where they will spend a considerable amount of time as they mature. But before they get there, the transit time is actually a 12-day journey for the spermatids. On this journey towards the epididymis, these spermatids will gain motility, and they will also gain fertilization capabilities. On to our last slide here. This is also a highly regulated hormonal process, which is these sperms are undergoing here. Gonadotropin releasing hormone, which is a hormone produced by the hypothalamus within the cranium, stimulates the anterior pituitary gland. And once stimulated, the anterior pituitary gland will produce luteinizing hormone, better known as LH, and follicle stimulating hormone, also known as FSH, to abbreviate these two hormones, important regulatory hormones. As the LH and the FSH are produced and released via the hypophyseal portal system, they eventually circulate to the testes for hormonal influence on Leydig cells and also Sertoli cells, respectively. FSH will promote paracrine molecule synthesis necessary for spermatogonia mitosis and spermatogenesis. FSH will also stimulate ABP and inhibin. On the luteinizing hormone side of the regulation, LH will target interstitial Leydig cells, and these cells, as we all know, produces testosterone, which is, a hormone. As the testosterone levels rise, they provide negative feedback back to the hypothalamus, thus inhibiting GnRH. So, in addition to the slides here, I did make another flow chart. This flow chart represents the hormonal regulation. And as I just spoke about, it starts in the hypothalamus, where the gonadotropin releasing hormone stimulates the anterior pituitary with our two very important regulatory hormones, FSH and LH, and their targets. So I hope this video presentation helped to explain the process, or the regulatory process, of sperm genesis. And next up will be the final exam. Thank you, and thank you for the semester, and see you later.